So Dr. James White is with us. He is from the plant biology department at Rutgers University. He is a professor and a researcher and maybe a budding friend of ours. We all experienced an earthquake together in New Jersey at our last meeting. Really, Dr. White experienced it, and Yazzie and I were on the computer, but it could have been a bonding exercise. Um, anyways, so he's with us. We're super excited to have him. He's been doing research with Enlightened Soil Core for 2 phases of a study now that we have shared with you all, and we'll continue that conversation throughout this webinar and afterwards. So please feel free to ask questions, and we invite your curiosity and your interest. And with that, I will turn it over to you, Dr. White.
Okay, thank you so much, Camille. I'm going to share my screen and gonna actually have a cat here that's trying to get my attention. I'm trying to get. Okay, sorry. Okay, so what I'm going to talk about is a number of things. Give some basic information about how plants use soil microbes and in this case, algae, green algae. But the process, you can see the title here. I'm going to talk about rhizophagy cycle. Rhizophagy is basically a term, refers to root phagy eating, root rhizophagy root eating cycle. And this is the process whereby plants will absorb microbes into the root cells and degrade them or expose them to reactive oxygen to get nutrients out of them. Some of them will be completely degraded. I'll also talk about endophytic microbes, endophytic, endophytes, internal phytic or phyte for plant. Basically, these are microbes that go inside plants. And both endophytes and rhizophagy cycle is affects plant development. And plants really don't develop properly without having those microbes in their cells and tissues, and I'll talk about that stuff. So a little bit about agriculture. We have 2 basic ways of doing agriculture. One of them is chemical inputs, or that is chemical agriculture. This is the conventional approach to agriculture. This is where we try to grow crops without microbiology, basically by putting fertilizers and other agrochemicals onto the crops in order to grow them. Okay, and the other approach is a biological approach, and this is where we use biology. And biology, they're bacteria, they're algae, they're algal extracts, there may be some sugars or some molasses people use, but basically they're fertilizing the microbes in the soil in order to get a plant response. So it's one uses chemistry to grow plants, the chemical agriculture conventional approach, and one uses microbes or stimulates microbiology either in the soil or in the plant to grow them. The chemical agriculture, of course, is not highly efficient. I mean, in the sense that a lot of the chemicals end up in the environment and do damage in the environment. Biological agriculture is somewhat more efficient in the sense that we don't have chemicals going out of the plant, off the plant, into the environment, doing damage to the environment. With biological agriculture, there's a lot of recycling. Plant materials are recycled and left in the soil and so forth. Biological agriculture results in plant products that are higher in nutrients than those produced with chemistry, with chemical inputs. In general, their crops are about 30% more dense in terms of nutrients than conventionally grown crops. And of course, most agriculture is somewhere in the middle between those 2 approaches. somewhere in the middle between chemical and biological agriculture. And the closer we get to biological agriculture, the cleaner it is, and the 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 safer the food supply is, fewer chemicals on it, and cleaner the environment is, better the foods are. This is a diagram that kind of illustrates, okay, to the left is a plant cells, chemical agriculture seeks to put the nutrients, the fertilizers as chemicals into the plant. But you can see I have um, a little funnel there to put those nutrients in just to kind of illustrate, but most of them don't go in, most of them go out. Chemical agriculture is only 30 to 40% efficient. Most of the chemicals go into the environment rather than in the plant. Biological agriculture relies on endophytic microbes, on endophytes. And really, this is a different system whereby plants absorb the microbes into their cells and then juice those microbes for nutrients inside their cells, actually inside their cells. And it's just my diagram here illustrates that. You can see the bacteria inside a plant cell. This is nearly 100% efficient because very little of the nutrients that are produced in the plant or that are extracted inside the plant go outside the plant. Very little of it go back to the soil. A little bit will go into the soil, but not very much. Most of it stays in. Okay, so first I'm going to talk about endophytes. An endophyte is any microbe that goes in plant tissues and does not cause disease. And you may be familiar with endophytes from the grasses, from things like fescue grass and ryegrass. They have fungal endophytes, and this is actually one of these. You can see this is a slide here. You can see some cells in the background. This is from a grass leaf or a grass stem. And you can see the blue line there. That's the fungus, the hypha. That's the endophytic fungus that's inside that plant. Okay, that's a fungal endophyte. But plants have many, many bacterial endophytes in addition to fungal endophytes. And this one is a specific kind from a tall fescue plant. But all plants have all kinds of endophytes. Okay, this shows a bacterial endophyte. This actually is a uh, is a is a a root tip. You can't see the root itself, but you can see some cells of that root, and you can see inside it there are these little clusters of blue things. Those actually are bacteria. Those are the bacteria belong to Micrococcus. Micrococcus, that's a bacterium that we actually pulled it out of tomato and we put it in this other plant. This is one of our test plants that you're looking at now. This is Rumex crispus or curly dock, a common weed that we took the seed, we sterilized the surface of it, got all the microbes off, and then we put this micrococcus on it. And you can see when the seed germinated, it went out and it took these microbes into the root. You can see these little blue dots there. Those are the bacteria that were taken into those root cells. So I'm just showing you this to show that what it looks like with the bacterium now inside those cells. What are these microbes? These microbes are, for one thing, they're all soil microbes. So plants get them from the soil. The first source of all the endophytes, primary of almost all of the endophytes, the first source is the soil. They come from the soil. And then the plant will get some of those endophytes that are really good, some of those microbes that are good, and put it in the seeds. And then they'll vector them in the seed. And of course, that's important because when the mama plant makes the seeds, it puts the endophytes or bacteria and fungi that those plants will need, that seedling will need, it puts those in that seed, on the seed and in the seed. And when that seed germinates, the little embryo comes out, it already has microbes that it needs to survive. And so that comes from the maternal parent. And I should say, we've been doing some work with corn lately, and we're finding that some of the microbes are actually vectored inside the pollen. So not only are the endophytes seed vectored, but they also are pollen vectored. And so plants could move their endophytes between plants through the pollen and the seed. So this actually is a grass seed, but I illustrating it to show that there actually are tissues. I'm not showing them. Well, you don't see them closely here, but the little leaf-like tissues on seeds, those they're called paleas and lemmas, but they're tissues that are on the seeds. They actually carry bacteria. They carry microbes, and when the seed germinates, those microbes come out and colonize those that embryo, that seedling. Okay, this is tomato. Tomato, we consider, uh, most people consider tomatoes to be sterile inside. If you cut them, you expect not to find microbes, but in fact, that's not correct. If you look in the tomato seed, around the seeds, around the cells around the seeds, there's going to be bacteria in there. And actually inside the seeds, there will be bacteria, little biofilms in there with bacteria in there. So the seeds will carry those bacteria with them. So the point being that healthy to keep to grow healthy plants, you actually need to have seeds that have their microbes on them. So you don't want to sterilize your seeds. That'll mess up the seedling. It'll actually cause seedlings to be more uh, susceptible to diseases like damping off disease and any other disease that comes along, because the bacteria actually are part of the immune system of the plant. So first, I'm going to talk about the rhizophagy process and how that works. And it actually was discovered in Australia, in Queensland, Australia. And these are two of the investigators that discovered it. But you could see this is a figure from their paper. And down here at E, you could see a root hair, and you can see little green dots in it. And what this is yeasts that these investigators labeled with green fluorescent protein, and then they fed their yeast to tomato plants, and those tomato plants took them in, and now you can see them inside the root hair there. Some of these other images show very similar things, but with bacteria. They labeled their bacteria with green fluorescent protein, and they tracked it into the plant. They published this paper in a journal called PLOS One, which is one of the open access journals that we use in biology and other, well, mostly biology. But they entitled this paper "Turning the Table: Plants Consume Microbes as a Source of Nutrients," that as opposed to microbes consume plants as a source of nutrients. So it's turned the table on that. They later named this process rhizophagy. About 2 years later, this paper was 2010. So 2 years later, around 2012, 2013, I guess 2013, something like that, they published a chapter in which they named the process rhizophagy, root eating, rhizophagy, root eating. And what we have been able to show is that it's not just bacteria and fungi. Yes, bacteria, you can see bacteria. This kind of shows the bacteria here. And you can see bacteria in it. Here, if you look below, that's a root hair. See the bacteria in it. That these bacteria would be invisible, except that we use stains for reactive oxygen. This particular one is for hydrogen peroxide. As it turns out, when these bacteria are inside root cells, the root is interacting with them using reactive oxygen, using superoxide is one of the main forms of reactive oxygen that the plant uses. But that makes it so you can see the bacteria, so you can see them inside there. And the middle one here, you can see this is actually a root hair that has a yeast in it. And you can see the yeast, these little dots in there, these are protoplast phases. When these yeasts or bacteria are inside root cells, they don't have cell walls. They exist as protoplasts, as naked protoplasts. So those little spherical things, those are those protoplasts. and you can see them coming out. They come out of the tips. They're actually ejected from the tips of these hairs, but you see the little brown there, and you can see small cells here as they first come out, and you see as they get older, they get bigger, they grow. That's typically how it works. In fact, the plant puts a little bit of exudates out there with that to make those for those bacteria, in this case the yeast, to regrow their cell walls and then go back into the soil and do their yeasty things in the soil, grow in the soil, gather more nutrients actually. Okay, this is a soil alga. This is actually the in-soil chlorella, and you can see there's some bacteria around it. You can see the bacteria there. There's 3 cells making that chlorella. This is what it looks like when it's inside a hair. This is a root hair, and you can see the chlorella. You can see it in there. It's actually losing chlorophyll. It's getting white. The blue in this case is superoxide. The plant has detected the alga in its cell, and it's hitting them with superoxide, and that's causing the loss of chlorophyll from this cell, from this alga. And it's also causing bacteria to come out of that alga. And you can see these little dots around it. Those are bacteria that are coming out of that alga. These algae will carry bacteria when they're absorbed in at the root tips, and I'll explain how that happens. They're absorbed in. Some of those bacteria go into the plant, and the plant gets those endophytes. So it's from one being an endophyte in the alga to being an endophyte in the plant. This is rhizophagy cycle. One of the things that we've shown at Rutgers is that the rhizophagy cycle is exactly that. It's a cycle. It's cyclic. It's not a one-way trip in. Instead, what happens is if you look at the root tip over here, this is a root, an actual root. You can see to the left here is where the root hairs are, the root hair formation zone, and to the right over here is where the meristem is. And this is actually over here is where the plant is secreting exudates, sugars, and sometimes some proteins, some vitamins, some amino acids also may be secreted, but those are attractants. Those exudates are attractants to microbes. And so microbes in the soil detect those exudates, and they go swimming towards the location of where those exudates are, and they find these root tips. And then the plant cultivates them at these tips, at the root tips, and takes them actually into the cells at this tip. Then in this zone, hits them with superoxide. Superoxide, which the plant can get from oxygen, makes from oxygen in the air, makes this highly reactive form of oxygen, which is superoxide, and oxidizes the cell walls off of those bacteria, off of those microbes, whatever they are, or green algae, whatever they are. They're oxidize the cell walls off of them and extracts them from nutrient, gets nutrients out of them. Then the surviving microbes, some of those microbes get completely broke down, completely degraded. We think a lot of the algae that get taken in are fully degraded, although some appear to be go all the way through the hairs, go through the plant and out the tip of the hairs. But if you look at the zone here, where the hairs are, you can see at the tips of the hairs, those little balls there. Those are in this case, those are bacteria that are being ejected out of the tips of the hairs. When those hairs grow, bacteria, microbes get ejected from the tip. And I'll explain how that happens. It's an interesting process. But as those hairs grow, microbes in the hairs are replicated, and then they're ejected back to the soil. So they go back into the soil, and they'll swim in the soil, get more nutrients, and then later to be attracted back. And so that's the cycle. That's how the cycle works. And this is just a diagram made by Harriet Mella in Austria, just showing that rhizophagy cycle. I'm not gonna. That's just a little cartoon of it. So you see the moving one, but this here shows now parts of the plant that are involved. If you look at here to the left, you can see this is a little grass root. You can see the root hairs there, and you can see the tip, and you can see around it the halo of bacteria. All of it, those are bacteria that are being cultivated by that plant at the root tip. That's how they work. That's what's going on here with roots. They're actually cultivating microbes, attracting, cultivating, and internalizing. And you can see where they're coming out. Here's the root hairs. You can see the tip of the hair. You can see that this is the bacteria now are stained with a nucleic acid stain, so you can see them, but you can see the little green dots coming out. Those are the bacteria that were ejected here to the, there's another one of the root hairs. You can see the little dots inside. Those are the protoplasts. Those are the bacterial protoplasts in there. These would be invisible if we didn't have that reactive oxygen stain. You can see now bacteria coming out the tip. They grow when they come out of the tips of those hairs. I should mention also, there are two places on a root where the cell walls are thin. One of them is right here at the root tip, where the plants take in bacteria and fungi or algae, whatever they are, they take them in there. That's where the cell walls are thin. And at the tip of the root hairs, those are thin, they're not hardened, so that things, microbes can go through the walls in both of those places. Okay. But it's a one way, they go in the tip and out in the root tip and out the root hair tip. That's how it works. This is actually a hair showing some of the chlorella. And you can see root hair, and you can see the chlorella, the cells here accumulating at the tip. And we think they came out in this case. These are living. And in this case, they actually went through the hair and ejected. Most of them don't appear to go through. Most of them appear to be degraded, but some of them are going all the way through. They seem to be going through those hairs. Okay, so going through the root. There's another one, shows that. You can see those bacteria accumulating, growing at the tip. You can see some of them are small too, so they're not giant. They're bigger, much bigger than bacteria, but they're not totally giant. They're they're uh, they have a small stage. So what is the rhizophagy cycle about? Well, for one thing, it's about nutrients. That when we measure, we give plants, we give them microbes, and then we let them grow, and then we can measure compared where we did not give microbes. When they do have microbes, they accumulate much more of all kinds of nutrients. Okay, but um, it's also about development. Development is critical, and without microbes, plants can't develop properly. And the reason we think that the development is happening, why the microbes are stimulating development, it is because the microbes produce hormones. They produce ethylene and they produce nitric oxide. Both of those are plant hormones. And some nitrate will be formed when the nitric oxide is produced. But anyways, those substances stimulate plant growth. And so they'll stimulate plant root hair growth and trichome growth and so forth, and cell growth in general. And the other thing that microbes do is in a plant is they will cause the plant to produce antioxidants, things like phenolics and enzymes like superoxide dismutase and catalases, antioxidants that make the plant itself more resistant to stress, because all stress is oxidative stress. And so with the antioxidants there, plants are hardier. They can tolerate more stress because of those antioxidants, especially the phenolics. They have a big effect on stress, making plants hardier. Also tastier, by the way, because those are also nutrients that people eat that are good for people. So this is a root tip, and you can see the bacteria accumulating there at the tip. See them there? And the plant is taking them in right at that zone, right in there. And they go in, this shows them. I'm This is stained for where the bacteria are in the root. This is a root. You can see this zone here. You can see the little blue dots. Those are bacteria going in. You can see to the left of that, this is older. They're losing their cell walls. All this irregular shape stuff there, those are the cell walls coming off. That's what happens when the plant hits these microbes with superoxide. The cell walls come off. Turns out the microbes carry nutrients on those cell walls. This just shows cell walls coming off again, coming off inside the root, inside those root cells. So the plant is getting those nutrients. So there's like metals, manganese, magnesium, calcium, different nutrients, even nitrogen are on those cell walls in those bacteria. There are actually places where those nutrients can attach. This shows the bacteria inside a root cell, and you can see they're living in there, they're growing, they're in a chain. You can see the smallest one there, and you see an older one. Okay, these bacteria are having to replicate as fast as they can in order to stay alive. If they don't, the plant will degrade them completely. So you see it's like a race to survive inside these plant cells. Okay, this is something my student Celeste Zong did, but she labeled these bacteria and then she, with something called, was actually M. cherry, which is a little label that you could put on the bacterium. And when it expresses, you can see this shows this red dye, this red fluorescent dye. And then she examined this with the confocal microscope, and you can see all the bacteria inside those cells, all the orange in there are the bacteria. So the bacteria go in these cells, and the other thing, they get into the cell. The other thing that will happen is the plant will move them around, and I'll show you an image that shows that. I don't want to I don't want to go too long because I want to leave time for questions, but I want to show a couple of these things. So this is what happens to the bacteria inside. And bacteria normally exist as rods or some other shape, like you see to the left. But once the plant gets them with reactive oxygen, the cell walls are removed, then they have these protoplasts. You can see these little spherical things. You can see they're all sizes and everything. They're the rules don't apply. Cell division doesn't take 20 minutes. Cell division takes just an instant, and you see they divide like that. This is what happens when they're inside the cell. This is this is this is how they grow inside the cell. These also are called L forms. I call them protoplasts, but there's a technical name for them when they're bacteria. They call them L forms. When they're fungi, they call them mycosomes. But these have been known already. This is not anything we invented. People have known this. It's just that people didn't know how to find them inside plant cells. They didn't have the stains. They didn't have the way to find them. So if we remove all microbes from a seed, then what happens when that seed germinates? You get a root that comes out, but you don't get any root hairs on it. No hairs will form. The other thing is that the gravitropic response is messed up. With no bacteria, roots normally will grow down, but with no bacteria, they don't. They stay on the surface or they go in the air even. They don't know where down is. You also get more root branching, you get more root elongation, you get bigger shoot. Big effects are in the roots if you don't have microbes there. Major effects. Roots can be tiny little things if you remove all the microbes. Here actually is a grass where we remove the microbes and we germinated it, and you can see there's no hairs on that root. The little bumps there, those are root hair initials, but there's no hairs there. This is what happens when you put a microbe back on. This is so we removed it like we did on the other one, and then we put a bacterium on it. This is a Pseudomonas bacterium, and you can see the hairs form right away. The hairs have to have bacteria on them to form. Here you can see the hairs. I don't mean on them, I mean in them. You can see these hairs here. You can see the little brown dots there. Those are the bacteria that are inside those hairs. And they're Without the bacteria there, you get no hairs. Here's what it looks like close up. You can see the hair, root hair, and you can see the bacteria in it all over. They're loaded with hairs, with bacteria. Here, actually is a little sedge growing. This was in Bonaire on the island. I did some research there years ago, but this sedge grows in these little coral limestone deposits. Doesn't have any soil, but it has sand and it has lots of microbes. And if we look at it, okay, look, here's a hair now. And you can see the brown areas are where the bacteria are inside that hair. And now above is a living hair. This is, now you can see how the hairs work, how these root hairs work. They move these microbes around. That's how they work. That's how they function. They're moving microbes. These root hairs are microbe processing structures. We think of them as absorptive structures, and they might be absorptive, but they certainly, for sure, they're microbe processing, and I'll explain that. I'll explain that. But you see these shadows of the bacteria going around and around. And what they do is, look, this is what happens, is that motion causes those bacterial protoplasts, those L forms, to break up. And you can see clusters of them right now. You can see some are old and some are young. Little ones that are darker are younger ones. The bigger ones that are clear are older and being degraded. Okay, but you see the movement like that causes them to break up apart into multiple. So the plant, by moving them, is making copies of those bacteria. So it's taking a few in at the tip and then making a lot of copies, getting nutrients out of them, then making a lot of copies inside and then putting those out in the, okay, and here, this is what it looks like. This is that hair. You can see it going around. You can see it going around and look at it. You can see the little white things going. Those are the bacteria. And it's depositing them at the tip. You can see them bumbling around at the tip. In mass, they secrete growth hormone, ethylene and nitric oxide, and that causes this to grow, this hair to grow. And when it grows, it shoots those bacteria that are at the tip. It shoots them out shoots them out of pores into the soil, and I'll show you that into the outside the tip. You can see that. This is a fat hair. You can see how fat it is. That's all bacteria in there. That's fat, fat, fat. They get fat with bacteria. These hairs get fat with bacteria. Yeah, sure, they're absorbing stuff, but they're mostly putting stuff out. They're mostly ejecting these bacteria. And here you can see bacteria that came out. All right. Here again, this is another plant, but you can see the root hair tip. Root hair, this blue in there is the bacteria in that tip. You can see some little spherical things. Those are where the pores are, where the bacteria come out. You can see the pores actually, and you see the bacteria on the outside where they were ejected, just to show that. And this actually is this is a little diagram that just shows that, but it basically what I'm trying to show here is that every growth spurt that the hair makes, bacteria are ejected out like you see there through little pits. And then the remaining bacteria that didn't get ejected are cycled around and increased in number, and then they accumulate in the tip again. And then in bulk again, in mass, they secrete those hormones causing growth spurt again, like that. Like that. This is an actual hair that shows a root hair that shows the bacteria in clusters. These bacteria didn't get ejected, they remained in. So you could see the kind of striped pattern where they stuck in there after where they accumulated, and you had a growth spurt and they did not get ejected. They stayed in there, but you could see that like that. So you see this peculiar banding pattern. This is how root hairs grow. They all grow like this. Every hair you get. Why didn't we find this later? Who knows? It's because this was invisible, because we didn't think that plants were doing anything like this. This is a tomato showing ejection. You can see the root hair tip, and you can see actually there's 3 pores down here. There's little pores down there coming where the bacteria are going out. So that's it. And then a little exudate comes out with this, and these would reform their cell walls and go out into the soil and acquire new nutrients. Okay, and I've already talked about this. Why is root hair growth linked to presence of intracellular bacteria? It's because they make hormones. That's why. And those hormones I've already talked about, ethylene, nitric oxide, and some of that turns into nitrate when it's oxidized, by the way. And so you'll get nitrate in there. And this is a quick summary slide. So what are plants getting from this? They're getting nutrients for one thing. And I didn't go over the data, but we've got different types of data that suggests that every microbe is different in terms of nutrients that it carries to the plant, because plants get a whole community of microbes. They take them in, they can get a lot of different nutrients, and they can essentially determine what microbes take in based on what nutrients they need. We don't know all the details of that, but apparently it appears that plants can do that. And based on some work that others have done. Increased oxidative stress tolerance in plants when plants are doing microbes, when they have lots of microbes, when they absorb those microbes, they get nutrients from microbes, when they're doing rhizophagy cycle, they're going to be more stress hardy. They're going to be able to take heat, and they're going to be able to take salt. They're going to be able to take some trampling or whatever. They're going to be more resistant to stress, because all stress is oxidative stress. Finally, and I didn't talk about this, these bacteria actually will go into the soil and colonize potentially pathogenic fungi and reduce their virulence, cause them to be non-virulent. So really the bacteria are part of the immune system of the plant. And so these microbes in general, including fungi and probably including the algae, are part of the immune system of the plant. So this is just a popular book. Jeff Lowenfels wrote this book called Teeming with Bacteria, Organic Gardener's Guide to Endophytic Bacteria and the Rhizophagy Cycle by Jeff Lowenfels, which describes this whole process. And I think that is all I have to show. And we can see if I can.
Thank you so much, Dr. White.
You're welcome.
We are going to take questions. So feel free to type anything into the chat. You can chat the whole group. You can chat me or Yazzie. Raise your hand as Ed is doing, or unmute yourself. So Ed, let's start with you.
Yeah, thanks, Dr. White. I'm wondering, so if you understood now how much of a plant can get nutrients, how much of their nutrients they can get if they have like an ideal, healthy, aggregated, where the atmosphere can get down into the soil and it's got the carbon and it's got the exudates, and compared to something more conventional, that might have some compaction, doesn't get the atmosphere in, has more chemicals interrupting it. Obviously, they're not producing as much, but what are sort of the percentages difference we're finding from like a traditional conventional field and something that's really regenerative? And what kind of percentages of the plant's needs can the plants get through this cycle, do you think now? Thank you.
Yeah, that's a good question. It depends what you look at. If you look at plants in nature, right? Plants in the wild, they are doing rhizophagy cycle. They're affiliating with mycorrhizal fungi. They have endophytes in their tissues, in which certain types of cells are sequestering bacteria, like trichomes, some like glandular trichomes, for example. If they have endodermis in the plant, they'll have microbes in that endodermal tissue. So wild plants will get all their nutrients from biology, all their nutrients from biology. Regenerative agriculture is the beginning of pushing us back to biology, right? Beginning to go closer to biology. I mean, we're still pushing plants. We're still growing crops by pushing them. And those crops, many of them don't have the same endophytes that they had when they were wild. So we're on a journey to get closer to that biological cultivation method. And what are the actual numbers? I can't tell you, Ed. I can't tell. I can only tell you that it is not as good as wild, and it is not as bad as conventional. But and you'll be able to tell because people who are doing regenerative agriculture are trying to push down their usage of chemical fertilizers, and so they could. So what are we talking about? It depends on. I can't tell you ideal numbers. Any other question?
Hello, this is Randy. Hey, I was wondering, we inoculate our legumes with bacteria, and I was thinking using the algae plus the inoculant together would make it work better yet. Some of the new inoculants, they're really good, but they don't compete with a inoculant that was put on the market, I think, 15 years ago, that outcompetes the new stuff, but the new stuff is better if from what I've read. What's your thoughts?
What are you think? What new stuff are you thinking of? There's something in particular?
I like soybean inoculant. I can't remember the exact names or of the inoculants right now. But they say some of the newer ones are more efficient, but the old style inoculants just outcompete the new ones. Do you think you're getting a good start with using the algae in combination with those inoculants?
Well, I think it'd be worth a try. Try to combine them and see if you can improve the effect. Uh, in terms of the old versus the new, those microbes, those were good microbes.
Mhm.
Those old microbes were good. They worked really well. Uh, it's hard to say how people could improve on the microbe. I'm sorry to say, I don't know if the new ones are. Now there are some things coming along where people are trying to emphasize the nitrogen fixation part, and that's developing. You hear a lot about that now, and companies are trying to develop products that for nitrogen fixation. It's not quite there yet, to be honest, but um, there's enough effort going into it that I wouldn't be surprised if there will be something on the market before too long. Now, I doubt, there is no silver bullet in the sense that you can't find the magic microbe. None of these are magic, right? And plants, don't forget, plants can get these from the soil. So, I mean, what we're doing is we're giving them, when we give them a biostimulant microbe or a biostimulant of any kind, we're trying to give them a step up, something to get them going, right? To just start going. A lot of these biostimulant microbes, what they do actually is they'll stimulate rhizophagy cycle, and they don't necessarily stay in the plant. They just go, give it a kickstart, they go in, the plant starts to take in more microbes, and so then the cycle starts going faster. And and really one microbe is not magic. You know, it's not enough. One microbe is not enough. It has to get those other microbes. So it's really a matter of just starting a system, kickstarting a system. Even with the algae, the algae go in, and they deliver not only their nutrients that they have, they deliver the bacteria that then the plant can use that, but they do something else too. When they lose their chlorophyll inside, that triggers the plant to start trying to absorb more microbes. It's kind of a humic substance effect, and that is the plants, when they get humic substances, they start absorbing more microbes, they secreting more exudates, absorbing more microbes. Well, the same thing happens with these algae. They go in, the plants get their cell contents and the chlorophylls, and they start to absorb more microbes. So it's a matter of triggering plants. It really is a biostimulant type effect. So I don't know if that helped with that question.
I think
Randy?
so.
Okay.
How often would you put algae on a plant?
Well, it depends. To begin with, I would do it to get it started, but I'd have to, I'd want to see what kind of effect I had when I put it on a second time. I'd want to see, do I get another jump, right? So does it boost it again? So, I don't know. We haven't done any field work with algae at Rutgers. We've done predominantly laboratory work to show how it's working. So I can't really say, but um, I'd if you're going to put it on again, put it watch and make sure you have make sure it has a boost, make sure it's doing something. I'd put it at least once to begin with, and I don't know, ladies. Can you guys want to weigh in? Do you have any data on that?
Yeah, we recommend 3 applications a season. We have growers who apply twice a season. We have some growers that only apply once, and some people who apply 4 times or more. So it really varies, but we recommend 3 applications, and we do notice a boost in the field, and that is, that's observational data across lots of different growers around the country with different crops, who have noticed whether it's a shift in the color and you're seeing that chlorophyll be enhanced in the plant, or whether it's a shift in the rate of growth or a shift in the stress adaptation. We've seen a lot of observational data around people who are applying the algae before a predicted stress event like a drought or a flood or something like that. And those plants that have been sprayed with the algae have a much higher capacity to survive the stress event. And so I know you spoke a little bit earlier, Dr. White, about the stress adaptation in the plants. So that's an example, Randy, of something that we do see a change in after an additional or third application.
Thank you, Randy, for that question.
Thank you.
You're
Dr.
welcome.
White?
Any other questions?
This is
Dr.
Jim.
White?
Hi, Jim.
Oh, I can hear you, Brad.
I have a question about plant sap. I squeezed some plant sap and looked at it under the microscope under epifluorescence, and of course the chlorophyll glows bright red, and it was coming from algae that looked like Chlorella vulgaris. Yet in the soil, I can't see it. Is there a propensity for it to show up in the plant sap?
That's interesting. Yeah, they're hard to see in the soil. It is true. Probably, I'm guessing probably because they may not be green. They may have a phase. If they're under the soil, they may have a phase where you're not able to see them. I agree with you. They're hard to see in the soil like that. In the plant, why are we seeing them in the plant? Probably because they're probably because there's some light there and you can pick them up. Are they're green because of the light. They also are easy to, without the soil around them and protozoans and everything around them, they also may be easier to see if you have just cell sap. You can pick them up. Some of those may be on the surface too, but they definitely are inside cells because, well, we see them in our experiments, but I have also done some work with some Europeans looking at some forest roots, forest trees, some of their native trees in Germany, and we've seen some interesting different kinds of algae inside those forest tree roots and the root hairs. So there's more than one kind of alga being absorbed. And why you can see it, I think it's probably just because there's less stuff to obscure it, and because there's light. If you're getting the plant sap, there's light there, and they're probably green, so you could see them.
Thank you.
You're welcome, Jim.
Brad, you want to ask your question?
Yes, Dr. White, I'm an agroecologist consultant down in Citrus in Florida, and I was wondering your thoughts on 80 to 90% of the citrus growers now are drilling holes in trees and injecting oxytetracycline to combat greening. And I'd like to know your thoughts on what that is doing to the rest of the biology in the tree.
Well, first off, I'll tell you, a certain amount of speculation. I'm going to be with citrus greening. I'm going to be speculating a little bit. Citrus greening is a microbiome disease, right? Probably what it is where the microbiome has shifted in one direction, and it's likely due to the use of chemical fertilizers on the plants. And so they've lost some microbes that normally would work nice, right? Instead, that's been replaced by the citrus greening microbes. And treating them with antibiotic, I mean, it's like treating humans with antibiotic, right? We're going to knock some more stuff out of the system. It's actually not the solution. The solution would be go back into the woods where we didn't treat those plants and get those trees that escaped, and repopulate those orchards and try it a new way. Go the biological way this time. But it's kind of hard with all those trees that are infected now. I don't know what to say. I don't think it's a good solution with the tetracycline antibiotic into the. Now they don't have an option, I'm sure. But it's not a good. It wouldn't be a good solution for people either if we're going to survive on tetracycline. It would be terrible for our health.
No, sure, no. I was just curious as to what you thought might be happening to the other biology in the tree.
They're messing it up.
Sure.
It's one screw up after another, right?
Right.
So that's typically what we do until we figure it out.
Thank you.
You're welcome. Thank you. It was a really good
Hey,
question.
Brad.
Yes, ma'am.
Oh, sorry to interrupt. This is Meredith.
Hey, Meredith.
Just thinking this would be an interesting area of focus. How are you? Good to see you. But yeah, I was just thinking this would be for us to look at with the in soil. Some of the other genomics tests we balancing in the bacterial ratio, plus an increase in diversity out in those tests. So we've talked about it quite some time, really wanting to look at greening up disease and how to report. So yeah, let's talk about that offline after this call sometime this week. There's a space for it. Maybe some trial work.
Sure.
I think it could be beneficial. Mm-hmm.
Go for it, Doug.
Dr. White, how are you today?
I'm good. I'm
Is
good.
this citrus greening, is that not the same effect that the farming industry today is headed towards if we don't start getting into the bacterial world and eliminating putting the salt fertilizer and the chemicals and the fungicides and everything else on our soil? Are we not heading to the same scenario?
Yeah, more than likely we are. More than likely. But that's only after we've destroyed our environment. Yeah.
Yeah. Why does it take so much? I speak to a lot of farmers all the time. Why does it take so much for them to understand that they cannot continue to put on salt fertilizers and chemicals, that they're going down a slippery slope?
Well, I think the reason is because it's really confusing. You know, you've got people telling you, and I mean, I studied agronomy like everybody else in agriculture, right? I had agronomy when I was an undergraduate, and I know what is in agronomy and what's taught. We all took that as fact, and
But
that's
it
how
wasn't.
that's how things worked. We thought, that's how they work. You put the chemicals on, and they absorb them, and da 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 da. So we have a lot of people that are still essentially believing and thinking and believing and saying the same old thing. And maybe they modify it a little bit with, okay, you could try a biostimulant microbe, but it's a snake oil or something like that.
Exactly.
It's they because the message is not out completely. That's the reason. And I think it's easy to ignore people saying it's still easy to ignore people saying that the biology is important.
It's
I think
Stockholm syndrome.
there you are, Liz. Yes. Yes. Yes.
Hi, Liz.
Yes.
Hi.
Yeah. Yeah. So it's like the Stockholm syndrome, right? They come to believe the captors, and the captors, I will say, I will put it in a way, the captors are uh is our old concepts, right? Those are the captors, our old concepts. I'm not going to blame big ag because we're all trying to do some good, and they were trying to do some good in producing all the chemicals and all the things that they did, but it ends up being a screw up. But like a lot of things, right? So yeah,
Thank you, Dr. White.
no, you're so welcome, Doug.
Thank you, everyone. We are at time here, so we want to wish everybody a great night. And big thanks to you, Dr. White, for joining us and sharing your knowledge and answering everyone's questions. We are always happy and interested to chat with anyone after the webinar, so please feel free to reach out to myself or Yazzie or Meredith or anyone on the team, and we would love to chat with you. Our next webinar with Donna will be on May 22nd. She's coming from Heifer International, so look out for that in the follow-up email, and we will be sending out the recording tomorrow. If anyone has any last questions for myself or Yazzie, we'll hang out for a second. Otherwise, have a great night, everyone. And thank you again, Dr. White.
You're so welcome. Y'all have a good night.
You
Thanks,
too.
Dr. White.
Bye.
Sleep well.
Thank you.